Humans always love to modify the world around them. This human curiosity was the reason that we even tried changing the genetic composition of plants and animals to reinforce valuable traits in them. With time, we became very good at this, but our understanding of such processes was incomplete until we discovered deoxyribonucleic acid or DNA. The DNA acts like an instruction manual for our cells, which tell them how to act and form the foundation of our every trait. But the problem was that gene editing was complicated, expensive, and took a lot of time to do. All this changed when a new technology entered the stage, clustered regularly interspaced short palindromic repeats, or simply CRISPR. Immediately, gene editing became faster, easier, and cheaper. But where did CRISPR come from? How does it even work? And what all can we do with it? Since the emergence of life, viruses and bacteria have been fighting each other. As the viruses need a host to survive, they try to insert the genetic code in bacteria and take over them. Most of the time, bacteria fails to resist because of their weak protection, but sometimes it is able to survive these attacks, and only then the bacteria is able to save a copy of the virus's DNA in its DNA library called CRISPR. When the virus attacks again, the bacteria quickly makes an RNA copy and arms a top secret weapon called Cas9. Cas9 scans the virus attacker by connecting every bit of DNA it finds in the DNA library, and when it finds a 100% perfect match, it is activated and cuts out the virus DNA, making it useless. But you might think, how does CRISPR help us in gene editing? Well, the breakthrough came when scientists figured out how to hack CRISPR. Here's how they use CRISPR in labs. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. The guide RNA directs Cas9 to the target gene and cuts out the DNA. Once the DNA is cut, the cell will try to repair it. Scientists also add a sequence of stencil DNA to their CRISPR so that the cell can perform an homology-directed repair. This stencil DNA is used as a blueprint for the rebuilding process, and in the end, we get a genetically modified DNA. Since its discovery, CRISPR has been used in various lab trials. Scientists were able to eliminate HIV virus from 50% of the living cells of a rat. Our worst enemy, cancer, could come to an end as CRISPR lets us edit our immune cells and make them better cancer hunters. Or we could possibly slow or reverse aging that can help us explore other planets or even the universe. But there are some downfalls of the technology too. Many fear that gene editing could be used to create perfect beings, which would be a threat to the current diverse human population. The technology is surely a bit frightening and raises big moral questions, but we have a lot to gain from it also. It's up to us to decide the best course forward, as CRISPR has the capability to change the future.